substances, complex mixtures of carbohydrates, lipids, etc. They can also sorb to mineral surfaces and bind minerals together. And this then forms all these large aggregates. It is really important to understand the different sizes that we have in a soil, starting from the clay to the silt and the sand-sized aggregates then. This is several orders of magnitude, and these associations in soil define a really complex and heterogeneous interface in the soil that is relevant for cation, anion exchange, but we also find hydrophobic surfaces that can also interact with other hydrophobic substances. It also defines the soil as a porous medium. As it is filled with air and water, we can have swelling, shrinking, control of water storage, control of water transport and of solutes. All this depends on how these aggregates form and how they look like. And these aggregates are also the habitat for the microorganisms that do all these organic matter transformations, and of course also for other microorganisms. So most of the organic matter in mineral soils is bound in aggregates, and this aggregation and the soil's carbon status is maintained by continuous feeding of the soil heterotrophic community with fixed carbon. And as we had pointed out, this is above ground and below ground litter, crop residues, and root exudates. And this means the type of land use and soil management is really decisive for carbon storage. How much organic matter we add to the soil and where it enters the soil. Now, my topic here will be mineral surfaces and organic matter accretion. I would like to look at the spatial distribution of organic matter at the microscale and on structure formation, aggregation, as it is determining soil functionality. The approach I want to use is, I would like to show some experiments and really well-designed, targeted experiments where we look at specific aspects and we combine this with soil analysis at the landscape or plot scale. And we think that if we both combine, we can elucidate a bit better how organic matter forms and how this affects soil functionality. First of all, I would like to show an incubation experiment that we did with 15N and 13.3 labeled litter. We had a control soil and unlabeled litter, soil and labeled litter, and we incubated for 24 days under conventional conditions in the lab. And we afterwards fractionated the soil and looked for the C13 and 15N label. And we find that this label immediately enters the clay fraction, both for carbon, but what is important, immediately most of the nitrogen goes, which is most of it organic bound nitrogen. So the nitrogen-rich compounds enter mainly the clay fraction. Of course, there's also a little bit in the silt fraction, but most of it is in the clay fraction. We then looked at these samples from the incubation experiment with NanoSIMS, secondary ion mass spectrometry, and I will shortly explain because I will use such data also later on. What you can do with that is you use a primary ion. In our case, it's cesium, and you sputter away the surface of the sample and release secondary ions with different masses that are then separated. And so you can have, and it's sensitive to element and isotope mapping. So you can have 12C, but also 13C. Oxygen always is a very nice way to look at the mineral surfaces. Generally, you can have 14N and you can have 15N, and also the minerals like iron and aluminum. And it has quite a high lateral resolution. So when we look at the results from this experiment, we see that we have only some single mineral particles, but mainly mineral clusters, like you see here. This is a scanning electron microscope image, and here the same spot in the NanoSIMS then. And what you see is most of the material is in clusters, and this is now blue, the oxygen, which is the mineral surfaces again, as you see, and in yellow, the carbon and nitrogen spots. So we find a very heterogeneous distribution of organic matter, and only about 20% of the visible mineral particles are covered by organic matter. This is estimated, these are just examples, this is estimated from a much larger number of regions of interest. Here again the same, and here we now look at the label of 12C 15N over 14N. So this is the enrichment with nitrogen-rich compounds, and in green, the enrichment with C13-rich compounds. So we see that these materials accumulate also in a very heterogeneous manner. So we have the old organic matter associations, like here, and we have the newly formed organo-mineral clusters. Still, the mineral surfaces remain partly free and only partly covered. Now to a field study. This is the retreat of a glacier in the Alps in Switzerland that has retreated since around 150 years. And we have thus a chronosequence of younger soils in the foreland of the glacier, and we have really quite old soils that have undergone a soil formation, let's say, for around 150 years. And we also looked at mature sites outside the glacier-covered area. We again did NanoSIMS with that, and this is a this is a combined image of 16O, 12C in green, and the CN in blue. And you see there's parts where this accumulates. We did again this suspension on silicon wafers, and then put this in the SEM and in the NanoSIMS, and did a number of measurements, and we calculated the coverage of the mineral surfaces. This is shown here in green from the NanoSIMS data. So we get a degree of SOM coverage of the mineral surfaces, and we also calculate the SOM connectivity. This means if it's more patchy, then it's 0, and 1 if it's more connected. If we then look at the data, we generally see that in these soils, organic carbon concentrations increase over time. They increase mainly due to accumulation in microaggregates. This is the fraction, what we call here light fractions, 1.6 to 2.2 gram per cubic centimeter with time. So in this fraction, it becomes lighter with time, or more of the mineral fraction enters up in this light fraction because of association with organic matter. So we have an increase of the light fine fraction at the expense of the heavy fraction. And in the mature soils, this is quite low then. And you see the mass contribution to bulk soil. So we really see that most of it goes to the light fraction. This means the SOM accumulation uses up these freshly produced mineral surfaces that are provided during weathering. If we then look at the SOM coverage in a number of particle size classes, we don't need to go into detail, but we see that it increases with time and increases again in the mature soil. And so the coverage increases, but even after long-term soil formation, the mineral surfaces are only partly covered. And if we look here at the SOM connectivity, we see that in the more mature soils, this increases. So this is what we already saw in our incubation experiment and confirms this result. So there's a growth of these patchy organic matter structures that progresses from already existing SOM patches, and they become larger, and this means they have then with time a higher connectivity. And we think this has implications for understanding how and where organic matter accumulation in mineral soils occurs. When we then look at the samples that we investigated, this shows again how this patchy organic matter accumulation is seen in our measurements. And yeah, it's just to illustrate how the connectivity increases with age of the soils. But when we say organic matter always starts to accumulate at points where there is already organic matter, this would mean there can't be really growth. So I would like to refer to two very nice papers by Peter Kopitke from Australia. And he looked in an incubation experiment again on the accumulation of organic matter from lucerne or buffalo grass. This is the C13 label again, and this is the 15 nitrogen. And we see that specifically nitrogen-rich microbial products originating from the decomposition of these materials directly bound to the mineral surfaces that were also not previously associated with organic matter. And these new associations on previously organic matter-free mineral surfaces are specific for the nitrogen-rich compounds. So this experiment was 365 days, was much longer than our previous experiment that I showed. And I think it explains how microbes are attached in their habitat, but also grow as biofilms or produce nitrogen-rich extracellular polymeric substances that then expand in the soil structure on the mineral surfaces, but also, of course, in the pore system. So I think it's important, and I think this is something we need to look at much more in the future, how important the role of microbes and their habitat is in the exploration and covering of mineral surfaces. I would now again come to an experiment, to a field study that we did in a typical agricultural soil of southern Germany. Here is Munich. We investigated a cambisol on a research station of TUM, and its tertiary sediments covered with loess. It's a sandy siltic cambisol, and we had arable plots under long-term crop rotation. This is how the landscape looks here. This is the soil profile, and we looked at the plowed AP horizon. We, in this case, looked at intact aggregates that were embedded in resin. You see a number of samples, and I will show data then from this aggregate. This is light microscopy, but we also did scanning electron microscopy and also NanoSIMS then. This is this aggregate again, and we measured a regular grid of 42 spots. The yellow spots indicated here are just for visibility. They are much larger than the surface that we looked at with NanoSIMS, because this has looks at spots of 30 to 30 micron. So what we looked at, but in a number of 42 measurements on this grid, corresponds to about 1.2% of the polished surface. And we looked at carbon, oxygen, nitrogen, Si, aluminum, and iron. And we measured more than 11 million points and did get these data for the different elements, and did multivariate statistics on that. And then we could see how these elements occur together or do not occur together. And we did these cluster analysis, and finally we see that the gray that is shown in the later pictures is the pores, and we have particulate organic matter. So the pores, I should mention, the gray, this is the resin that can be differentiated. It's of course a carbon-rich material, but we can differentiate between the resin carbon and the organic matter carbon in soils. Particulate organic matter is rich in carbon and nitrogen. Then we have aluminum-rich mineral grains. We have Si-rich mineral grains that have more oxygen and Si. And we have aluminum-rich clays that have a lot of organic matter, and the same for Si clays. When we then look at all these 42 images, we were really not expecting this, but we don't see just a heterogeneous distribution, but we see two different types of what we call microdomains that are clearly different within this one aggregate that we have, are clearly different in their composition. And so these are, I should say, this is microdomain A here, where the blue dominates. We have a in between, sometimes microdomain B, but not very often, and microdomain C, where the yellow and green components dominate. And we see that in A, we have a lot of Si-rich mineral grains. In C, we have a lot of aluminum-rich minerals. The same for Si clays that dominate in A and aluminum clays that dominate in C. We have little iron oxides. We see that pores are much more in this A domain, and particulate organic matter is much more in the C domain. And when we look at the pore system, because this goes then in the direction of functionality, we see the number of pores, it's much higher in these A domains compared to the C domain. Also, the median of the single pore area is larger in A, and the maximum single pore diameter is much higher in the A domain compared to the C domain. If we try to get this together, we see that this microdomain A is having these large blue parts, and we think this is mainly quartz minerals, so sand or silt-sized quartz particles. They are surrounded by thin layers of phyllosilicates, most probably smectites, but it's thin layers that are around. Generally, these are low in organic matter, so there is some organic matter here, but it's only here in these parts, it's comparable low, and we have a lot of the gray spots here and here and here, and this is the large pores, so that we finally have a high total pore area. So this is how within the one aggregate this microdomain A looks like. The microdomain C looks completely different. It has these blue parts that are small, aluminum-rich, most probably primary minerals like feldspars, but they are in a phyllosilicate matrix. The yellow, this is most probably illite, and they are surrounded by, they are high in organic matter themselves, and they surround particulate organic matter, which is the green. So there is a lot of organic matter here, and this shows how the organic matter associates with these minerals and that surrounds that, and also the that surrounds these particulate organic matter residues. And this very much goes back to what Tistel and Oates in their classical paper in '82 have postulated, that the particulate organic matter is surrounded by minerals, and minerals that are glued together with organic matter. And the gray, we have only few and narrow pores and a small total pore area. And now this, of course, if you look how such an aggregate is built up with all these different spots occurring in near vicinity, we have a microdomain A that has a completely different functionality compared to this part of the aggregate. So here, the sorptive potential is accessible. We have large pores, water can penetrate and then diffuse to these mineral surfaces. The pores are large and habitable for microbes, and the organic matter is easily accessible, maybe also exchangeable here at the surfaces, because they are near the percolating water in the soil. This domain, in contrast, has generally a high sorptive potential, but that's less available because there's only small pores. These pores are so small that most of them are not available for microbes as habitat, and only chemical reactions occur here and sorption processes, but also diffusion in these small pores would be limited. And generally, the organic matter here in this domain seems to be protected from microbial attack because the microbes can't enter these small pores. So this shows how strongly organic matter and organic matter formed aggregates have different functional domains within one aggregate, and of course the same would occur in the next aggregate of this soil. So we think, to conclude, that the micro-architectural arrangement of soil components is like such a house of cards structure that is partly filled with organic matter in association with the minerals. This will define the microbial habitat also here, but it can accumulate more organic matter, and this also says we have organic matter associated with the mineral surfaces, but with time they fill also the pores. So the organic matter accumulates in soils, not completely independent of the mineral surface area, but we think the mineral surface area does not finally limit the accumulation of organic matter. And this would also mean that there's no limit predetermined by clay content or soil texture for the capacity of soils to sequester organic matter. From my point of view, this is much more depending on the input of organic matter. And this would explain why we, of course, find a relation between mineral surface area and organic matter accumulation in soils, but very often these relationships that we find in soils are quite weak, and other things, other parameters come in, and we think this is one explanation why this could be the case. And organic matter can accumulate and also be lost again, rather independent probably of the total mineral surface area. So we think that the observation of soils and their composition at the scale reported here, that is in the micrometer range in below, seems to be specifically relevant for understanding the emergent soil functional properties. We think these data show new perspectives for the potential of soils to sequester organic carbon and at the same time fulfill other ecosystem services, like storage of nutrients via cation exchange of the mineral surfaces. So the organic matter does not completely cover and also not completely interfere with these functional properties of the mineral surfaces. And we think what we need to understand is the intrinsic capability of clay-sized minerals and mineral mixtures in the soil to form such emergent soil aggregate structures, and also the dynamic change in the pore structures that are induced by, of course, physical processes, but also the microbial processing of organic matter. So we think soil type and mineral composition matter because they define beyond surface area the microstructural arrangement of organic matter and minerals and the microbial habitat. And with this, I would like to acknowledge our funding and thank you for listening.
Thank you, ma'am, for such a wonderful presentation and giving you valuable insights on this very important topic. So with that, we will go ahead and take some time for questions. So I will request my friend, Dr. Iqbal, if there are any questions.
Welcome you, Professor Ingrid. It was a nice presentation overall. We have some limited queries for this session. One important query is, somebody is asking what is the importance of organomineral complex in soil fertility improvement program?
Can you repeat the question?
Yes, ma'am. Yes, ma'am. Yes, ma'am. Yes, ma'am. Importance of organic mineral and complex in soil fertility improvement program.
It's very important. I think the structural arrangement really stabilizes the soil against erosion and also adds functionality to the soil. The organic matter, if it's added and does not completely cover the minerals stay available for nutrient binding, but also the exchange capacity of the organic matter is added to the soil.
Thank you, madam. Thank you, madam. So, yeah. One Dr. Shahida Nisar is asking, you said nitrogen-rich compounds form new organomineral complexes. I want to know what kind of N-rich compounds.
What I think it's basically proteinaceous type material and also amino sugars. Part of this organic matter that accumulates is microbial necromass, and microbes, their cell walls are formed by amino sugars. So this is a large part, and the rest, I think, it's proteins, oligopeptides, and things like that.
Madam, one question I was asking from on my behalf. Under our conditions, our developing nation conditions, we have limited resources regarding the research in organomineral complexes. We do not have big laboratories. We do not have X-ray diffractograms. That equipment is needed for these important mineral studies. Madam, how can we do these mineral studies on our limited resources? What we have already.
Yeah, of course, this is basic and research that is not immediately applicable. I think generally what you need to see for the status of the soil is to have carbon concentrations and stocks. I think this is, if you want to see then in what state your soil is, it's important to see how much carbon is stored, so a lot can be already done if you have a proper measurement of carbon concentrations, and if the sampling is done correctly. And for me, this means you also record bulk density, because then you can calculate carbon stocks. That's important. And you go also to the subsoil. I still see a lot of studies that only look at the topsoil, let's say the first 20 centimeters or so, and ignore the subsoil, which stores large carbon stocks, although the carbon concentrations are low. So to understand how soils contribute to carbon storage and also contribute to soil fertility, we should also look at the subsoil, because plants partly explore the subsoil also. And it's important to make sure that plants also have this resource and are not confined to the first 20 or 30 centimeters.
Thank you, Professor Ingrid, for your valuable presentation. Thank you.
Mm-hmm.
Thank you, Dr. Iqbal. Yeah, thank you, Dr. Iqbal, for sharing the questions with us. And I would like to thank Dr. Ingrid Kogel for such a nice answers and presentations. In fact, I think that this is one of the basic research where we can understand how the mineral and organic matter just interact with each other and stabilize organic matter in a longer period of time. And I think from this, we can understand a lot of things, how the mapping of soil organic matter at a nanoscale can be done using these sophisticated instruments like NanoSIMS and extra. So with this, we have come to our webinar.
Sorry, just to mention, we are an open facility. We accept researchers from all around the world if they want to measure with us. We have, in the present time, even developed a way that you don't need to come to our lab. If you are interested to doing things like this, you can take part in the measurements with a remote connection, and yeah, you are welcome to try to use our facility.
Well, that's wonderful. That sounds really great. I mean, we would like to have such collaborations with you. So if it's possible, could you please share all those informations with us so that we can put those queries to our participants if they have any and want to collaborate with you.
They can just look at our webpage. We have information there how it works.
Okay, that's great. Thank you. Yeah, so with this, we have come to an end of our webinar. I would like to thank everyone who made this webinar possible. And I especially thank Professor Ingrid for her wonderful presentation. And thank you once again for coming. Here also, I would like to remind our participants that we are having a second lecture that will start at 6:30, and Professor Ratan Lal will join us. So be available at that time. So thank you, Professor Ingrid, for joining us and sparing your valuable time, and in fact, accepting our invitation at such a short notice. Really, that was wonderful.
Thank you. And thank you.
Yeah, bye.
Bye.
Sorry, group photograph.
Yeah, yes. Sorry, I forgot. Before we end up, can please everyone please open your camera so that we can have a group photograph. Please open up your camera. Okay, thank you. We are done, and see you later.